Dear students, in the last video we have discussed about what are the requirement of therapeutic agents and vehicles in the preparation of parenterals. Today we will discuss about what are the requirements of various added substances or various additives. If required only, these additives are added. Suitable substance may be added to the preparations of injections to increase the stability or usefulness of the preparations. The amount of added substances should be harmless. That is very minimum amount we have to add, and it should not have any adverse drug reaction or side effects. Do not interfere with the therapeutic efficacy. There should not be any incompatibility or reactions. Even during the analysis, they should not interfere. Okay. So whenever you are carrying out the test analysis, this substance should not interfere or interact. And no coloring agent is added. Okay, we cannot color the parenteral preparations. Okay, and we are not adding any coloring agent. Sometimes injections have color because of the API. Okay, API itself is colored like cyanocobalamin. Then special care in the choice of and use of added substances in volume exceeding 5 ml dose. Whenever we are giving more than 5 ml of the dose per one dose, then it should not contain more amount of the added substances, because it may cause any toxicity once again. To prevent the growth of microorganisms, preservative must be added in multiple dose containers like diclofenac sodium injections. So one vial, from the one vial it is given to many patients. They are called multiple dose containers. Okay, for every patient 1 ml is given from that particular vial. So whenever you are removing from that vial, there is a contamination of the vial. So you have to, that is why we have to add that is preservatives to prevent the growth of microorganisms. Air in the container to be displaced with chemically inert gas due to sensitivity of drugs to oxygen. If the drug is sensitive to oxygen, we have to replace by inert gases like nitrogen or argon gas. Next, coming to one by one, first is antimicrobial agents or preservatives. They prevent the growth of microorganisms. They may be bactericidal or bacteriostatic in nature. So better to avoid the use of antimicrobial agent wherever possible. Sometimes this antimicrobial agent act with the API, active pharmaceutical ingredient, thereby affect the effect of the drug or preservative itself is decreased. Like thiomersal is a preservative, it will react with the proteins and the efficacy of the preservative itself is reduced. Preservatives are used in multiple dose parenteral products, already I told you, to prevent the accidental contamination while withdrawing the contents. Then antimicrobial agents should not be included in infusions, mainly in case of large volume parenterals, that is more than 100 ml. We should not use any antimicrobial agent. This because there is a chance of toxic dose of preservatives to the patient, because whenever we are giving, it's not one bottle, we are giving 3-4 bottles per day infusions or nutrients, there may be a chance of toxic dose by the preservatives itself. More and more amount of preservatives goes into the body, it will cause a toxic reaction. So examples for preservatives, that is benzyl alcohol, benzethonium chloride, methyl paraben, propyl paraben, phenol, thiomersal. So you can see they are in very minute quantities, many minute quantities, that is 0.5 to 1%, even the 0.01%, so much less the quantity in the case of injections. Next, what are the antioxidants added? Second one. So antioxidants means they prevent the oxidation of the product during the production or during the storage, used to protect the product from oxidation during storage and steam sterilization. There are 4 types depend upon the mechanism of action. First is reducing agents, acts as a reducing agent, thereby prevent the oxidation. Examples are ascorbic acid, sodium bisulfite, sodium metabisulfite, thiourea. These are reducing agents which acts as a antioxidants. Then B, blocking agents. They block the oxidative chain reaction. Examples are butylated hydroxytoluene and tocophenols. And synergistic action, that is enhance the effect of antioxidants. Examples are citric acid and tartaric acid. Last one is chelating agents. They form a complex with the substances which catalyze the oxidative reactions. Okay, example is EDTA, that is ethylene diamide tetraacetic acid. So these are the 4 types of antioxidants which prevent the oxidation of the product. Next, buffers. So to have a physical and chemical stability of the product, that's why we are adding buffers, added to maintain the desired pH, that is ideal pH 7.4. The buffers added are acetate buffers, citrate buffers, phosphate buffers usually used, which are compatible with the body fluids and even the products. So alteration in the pH in the parenteral product may occur due to degradation of therapeutic agents, incorporation of leached out of alkali from the glass container into the product, or release of acid or alkaline impurities into the product from the rubber closure. Okay, if there is release of such material or degradation of your therapeutic agent or release of any alkali from the container, or there is any from the closure also, any matter releases into the product, there is a change in the pH, it may harm the product. To prevent that, we have buffers, we are using buffers. So what are the factors affecting a selection of buffers? They should have effective range, and what is the buffer capacity, that is a concentration range, and chemical effect on the total product. Buffers are nothing but they are chemicals. When you add into the product, what is the effect on the product, that also we have to see. So examples are acetic acid buffer, citric acid buffer, lactic acid buffer, phosphate buffers. Widely used are phosphate buffers. They are used in the concentration of 0.1 to 5%. Next is chelating agents, used to form complex with the metallic ions present in the formulation so that the ions will not interfere in the manufacture of formulations, because during manufacturing we are using equipment, even the raw materials that may contain some heavy metals or metallic impurities. So this will complex with those and prevent the product from any oxidation and all. So that is why we are adding chelating agents. So example and get all. So examples are disodium editate, dicalcium editate and tetrasodium editate. These are the example for chelating agents. So they will form a complex with the metallic, which are from maybe from the equipment, from the raw material or from the packing material like rubber closures. So they form a complex and get dissolved in the product itself. Next is inert gases. So product stability and integrity of oxygen sensitive drugs can be improved. Some drugs which are sensitive to oxygen, so we have to remove the oxygen and we have to fill the gases like nitrogen and argon gas, otherwise the drug will undergo oxidation. It may be during the production operation also or during the storage of containers. Okay, examples of inert gases are nitrogen or argon gas. Then surfactants. Surfactants mainly used to enhance the solubility of drugs which are poorly soluble, or to disperse the drugs, water insoluble drugs, to form colloidal dispersion, mainly used in parenteral emulsion and suspension as wetting and solubilizing agents. So it should be compatible with the therapeutic agents. Widely used are propylene glycol, povidone, polysorbate 80. These are the various surfactants used to enhance the solubility and disperse the particles in case of emulsion and suspensions. Next, stabilizers. A parenterals are available in solutions and most prone are unstabilized. Okay, usually this liquid orals or liquid products, they are unstable when you compare to tablets or capsules. To enhance the stability of your formulation, we have to add stabilizers. Like example like creatinine, glycerin, niacinamide or sodium saccharin, sodium caprylate. So these are enhance the solubility. Okay, they are enhancing the stability of the products. Oh, okay, that's why they are called stabilizers. Next one is solubilizing agents. Even surfactants also used as a solubilizing agents. So to increase the solubility of slightly soluble drugs, they act any one of the following mechanisms. One is solubilizers, they have the co-solvency effect or emulsifier or acts as a wetting agents. Examples are dimethyl acetamide, ethanol, glycerine, PEG 400, that is polyethylene glycol 400, polyethylene glycol 300, when the polysorbate 20, 40, 80. So these are the different grades. So they act as a solubilizers. Okay, you dissolve the drug in small amount of these solvent and dilute with the diluent like WFI, okay, or vehicle. So thereby enhance the solubility. That is called co-solvent. Next is tonicity agents. So used to reduce the pain at the site of injection. Okay, if there is a hypertonic solution or hypotonic solution, there is a problem. The injection should be isotonic in nature. To have a tonicity say similar as that of the plasma or your blood, we have to add tonicity, isotonic agent. So buffer also may act as a tonicity contributors as well as stabilizers for the pH. So isotonicity depends upon the permeability of the leaving semi-permeable membrane. Okay, if you inject hypotonic solution, what happens? There is a problem. There is a swelling of cells occur and enlargement. That is called hypotonic solution. Means the concentration of the injection is below that of the your RBCs. What happens? There is a, when you inject such things, what happens? There is a swelling of the enlargement of the cells takes place. That is called hypotonic. Okay, it will take the solution or solvent from the your solution and they become enlarged. If you inject hypertonic, what happens? There is shrinking of cells, that is reduction of cells, RBC cells. It is called hypertonic solution. Means you are injecting more concentrated solution or isotonicity is more than your RBCs. Then there is a shrinkage of cells. You might have seen in your home, that is concentrated sodium chloride you are putting for mangoes, tender mangoes. What happens? If hypotonic solution or if the solution is hypertonic, the shrinking of tender mangoes. Similarly, if you put water into the grapes, dried grapes, what happens? There is a swelling of the grapes, that is hypotonic solution. That is how. Similar will occur in the case of RBCs also. Then, so these are the example for tonicities, that is dextrose 5%, sodium chloride 0.9%. We can use even the sorbitol, glycerin and mannitol. Next, last one, that is protectants. To prevent the loss of activity of therapeutic agents, what they will do during the preparation or processing, there are various steps: filtration, sterilization or filling. So that time what happens, this drug molecule or drugs API may stick into the product or stick into the container, or they may degrade during the processing. To prevent that, we are adding protectants. Loss of therapeutic activity may be due to stress caused during the manufacturing process, like freezing, drying, in lyophilization, mainly in case of protein drugs like liposomes, even the protein formulations, even the vaccines and all. So degradation. So adsorption of therapeutic agent during the processing, during filtration by equipment surface or container enclosures. Okay, then what happens if adsorption is there? So you should not, it won't go into the solution, entire drug won't go into the solution. So that is why we have to prevent that by using protecting agents. So human serum albumin acts as a protectant and prevent the adsorption of insulin hormone by itself being adsorbed. Whenever you are manufacturing insulin injection, what happens? If you add such insulin you are filtering, what happens? It will be adsorbed by the on the filter membrane. Okay, so that is why what we are doing, we are adding human serum albumin. Okay, so what happens when you add human serum albumin, it will get adsorbed rather than insulin, so there won't be any loss of product. So they are called, your protectant is human serum albumin for insulin drug. Next, there are two types we can divide, that is cryoprotectants. means it is based on the how process that name has given. Prevent the loss of activity of active ingredient due to freezing. Okay, there is a cooling down of the product, freezing, you are doing freezing. That time what happens? There may be loss of activity. Example, polyethylene glycol added to prevent the damage of enzyme lactate dehydrogenase and phosphofructokinase caused due to freezing. So whenever you freeze as such that these two enzymes, that is lactate dehydrogenase and phosphate fructokinase, what happens? There is a loss of activity. To prevent that, we are adding polyethylene glycol, so which acts as a protectant. Okay, so that is why that is called cryoprotectants. Next one is lyoprotectants. So here what we are freeze drying the product. There it is freezing. Here freeze drying one is a process. It is a process like similar to drying process. Here what? Cooling and evaporating. So that process takes place. That is why it is called freeze drying. Some drugs like biological hormones and all, you cannot dry in as such in the dryer. So to prevent that, we have to use free drying process. So which prevent the loss. So in this what happens? We are adding lyoprotectant to prevent the loss of activity of active ingredient. Okay, so examples are sucrose and trehalose, okay, which prevent the loss of drug. So that's about the additives or excipients used in the parenterals. This is very important question. 10 marks question will be asked in the university exam. What are the requirements or what are the additives added in the parenterals? Thank you.